come to the end of this chapter, which is structural organization in plant, and the last topic that is left is seed. So let us discuss about the seed, what it is and what are its different types. So what is seed? It is the matured ovule which happens after fertilization. After the process of fertilization has occurred in the plant, then the ovary will change into seed. Lot of chemical changes happen after the fertilization process has happened, and this chemical changes, it results in ovary turning into fruit and ovule turning into seed, where it has the embryo inside it, and this embryo, when it gets favorable condition, it will develop into a new plant. So that is what seed is, and the importance of seed is there, that it protects and it keeps the future plant safe and secure. It has so much of food that it can feed the dormant embryo up till the time favorable condition is met, wherein it can get food from outside, and then the process of development can begin. Up till that time, the seed is the thing which will keep the embryo alive and nourished, and it will keep it protected. So that is wherein lies the function of seed, and seed also helps in dispersal. So the two important function of seed that we get is function of seed, number one, to help in dispersal. Dispersal of plant, to go, like it is a seed with which the plant will go, so it helps in dispersal of plant. To help in the dispersal of plant, and secondly, function is to keep the embryo well protected, nourished, or well fed up till the favorable time comes. Then nature will itself take over the activity, the role of nurturing the future plant. The embryo will be nurtured by nature itself, isn't it? It will get enough oxygen, then air, water, everything, and it will grow into a new plant. But till that time, it is the seed which is the guardian of the embryo, which keeps the embryo intact and live. So that is the function of seed. Now let us quickly go into what are the different types and what are the different categories in which seeds are divided. So when we talk about seed, there are a few features inside the seed which we will have to consider. The first feature being how many leaves are there in the seed. How many leaves, and seed leaf are known as cotyledon. How many cotyledons are present in the seed? Based on this, seed is divided. And secondly, another point of classification of seed is the presence or absence of endosperm. So these two are the criteria on which seeds are divided. Now let us see, based on cotyledon, seed is divided into monocot, which has one cotyledon. Example is monocot example will be palm, grass, coconut, etc. Dicot. Dicot example will be gram, then P, then we have polycotyledon. These are all cotyledon. Add that, monocotyledon. I am not adding that because of restraint of place, but the word will be monocotyledon, dicotyledon, polycotyledon. Polycotyledon few. Some gymnosperm are polycotyledon like cycas, pinus. They possess many leaves in the seed. So that is based on cotyledon, number of cotyledon. Monocot will have one cotyledon, dicotyledon will have two cotyledons, and polycotyledons will have many cotyledons. Now we shall see in detail the structure of monocot seed and dicot seed. First let us talk about endosperm. Now endosperm is that part which in case of angiosperm, the endosperm is triploid. As we know, it results from double fertilization, and its purpose is to nourish the embryo. That is the purpose of endosperm. In case of gymnosperm, endosperm is haploid. But the thing is, some seed after development, they may have endosperm retained in them, or in the process of development, they have used the entire endosperm. So if they have used the entire endosperm, those seeds are known as X albuminous or non-endospermic seed. Basically, mostly dicots. Some monocots are also there, but it is mostly dicots which are X albuminous. Then those seeds in which an endosperm persists even after the development of the seed as a whole, they are known as albuminous. Albuminous seed or endospermic seed, and these are mostly monocots. I am not giving the example separately. In both these cases, there are exceptions. Like we have few monocots which are X-albuminous, and few dicots which are albuminous. I would suggest you please refer to the book to find out the example. It is provided in all the books. As of now, I'll just mention what are the classification and on what basis the classification has been done. So that is what. Now we will go to detailed structure of monocot seed and dicot seed, and let us try to find out how are the development of seed undertaking. So now I'll just try to do a rough diagram. And do the embryo with the help of So that's a basic diagram of dicot seed. Dicot seed. Now let us see the parts. These are the two cotyledons. Cotyledon. Now this is developing embryo, and this is the embryo. The lower portion. Let's mark this point. This part is the radicle, from where comes out future root. Radicle will give rise to the hypocotyl. This is radicle, which gives rise to hypocotyl. And this part is plumule, which gives rise to epicotyl. So this is the structure of a dicot seed. Now let us see monocot seed. There is not much place for monocot, so I will make a smaller one. So monocot seed, as I have already told you, it has endosperm even in mature state. So this is roughly a so this is monocot seed. Now let us quickly do the labeling. This portion, this is the endosperm. They have quite a good stock of endosperm in the matured seed also, which we will not see in dicot. Then this is the septum. This portion is cotyledon, which is, cotyledon here is much reduced and it is known as scutellum. S-C-U-T-E-L-L-U-M, scutellum. It is the cotyledon. Now here in case of monocot seed we have extra structure which are the protection of epicotyl and hypocotyl. They are known as respectively coleoptile, which is the protecting covering of epicotyl, which will give rise to the shoot. And we have here, this is hypocotyl, which will give rise to root, and the protecting covering of hypocotyl is known as coleorhiza. So we have coleoptile and coleorhiza in case of monocot seed, which were missing in the dicot seed. So now let us quickly just go through the difference between these two seeds. So from diagram only we can make it out. Dicot has got 2 cotyledon, monocot has got 1 cotyledon. Then in case of dicot there is no endosperm, but monocot has endosperm. In case of monocot, the cotyledon is reduced and it is known as scutellum. But here we have fully developed cotyledon. Then next important point will be monocot seed. It has coleoptile and coleorhiza. These are the protecting covering of epicotyl and hypocotyl. Coleoptile and coleorhiza are absent in case of dicot seed. So this is a very prominent difference between the two. So from here I think you can make 3 or 4 difference. Difference is what that is basically asked. So you should learn the diagram. So with this we come to the end of this chapter, which is chapter 4, structural organization of plants. I would just expect that you will do all the whatever board work I have done. I have mentioned all the important points, so please go through the video and note down all the points that have been told to you. Make note of it and practice the diagram as much as you can. Biology diagram is the most important thing. If you explain with the help of diagram, you will get the full credit. And you have to remember the examples wherever required and the difference whenever there are 2 things like here, monocot, dicot seed, then true fruit, false fruit, parthenocarpic fruit, true fruit. These kind of differences you will have to keep in mind. I would advise you to go through the video again and again, note down all the important points, and 2 things that you should keep in mind are examples. Wherever examples are there, examples are very important because questions are put on them, and differences. Differences are very important, as in case of this dicot, monocot that we did, true fruit, false fruit that we did. Those things are very important. And this is all, and I hope you prepare your chapter well. All the best. And we shall see each other now in the 5th chapter, that is structural organization of animals.